The human genome is one of the most fascinating and amazing features of biology. It consists of 3 billion nucleotide bases that contain the information for 30,000 protein-coding genes. And when a mistake or mutation occurs in the genome, it can have catastrophic effects on the cell. In fact, many cancers and diseases can be traced back to a single mutation. In an effort to treat such diseases, scientists employ a technique called gene therapy. Gene therapy is the use of DNA to treat disease. It's based on a very simple idea. If a mutation in DNA is causing a problem, then fix the mutation and you've fixed the problem. However, the execution of this idea is difficult. One field that is trying to perfect gene therapy is synthetic biology. Dr. Zhao is a research professor at the University of Illinois at Urbana-Champaign, who is using synthetic biology to create new tools for gene therapy.
Here, I think synthetic biology is a relatively young field, but it's a very rapidly growing field, and still there are many definitions for synthetic biology. It depends on who you talk to, right?
Mm-hmm.
So
Yeah.
there's no like a consensus definition. But in my lab, we typically define it as the design of biological systems using principles derived from engineering, chemistry, physics, and biology. The area we're interested in developing new tools for treatment of human health, right? One direct application of those synthetic biology tools is gene therapy.
Synthetic biology is a relatively new field that combines engineering and biology to create standardized and modular tools for genetic engineering. The modular nature of these tools means that they are easily interchanged and modified, making laboratory work faster. This new approach to genetic engineering is changing the older approaches taken to gene therapy.
I think gene therapy, of course, is a field, as you know, with lots of promises, but also lots of failures as well. Right? It was started more than like 3 decades ago, and then the
Yes.
first few trials that were very successful,
Mhm.
right?
Sure.
That's why people became very excited about the field. But then the more you tried, then also you saw more failures as well. But I think that the field is still very promising, clearly, and I think gene therapy is a very powerful tool to treat human disease, especially those genetic diseases.
Dr. Zhao's lab work focuses on creating gene switches using small molecules. When the gene switch is on, the small molecules go to work correcting mutated DNA. When the gene switch is off, the small molecules go away, and the mutated DNA is expressed as normal. Overall, the gene switch offers a great amount of control over when the gene therapy is administered. The hope is that this makes the therapy safer. The small molecule that they use is an artificial nuclease called the TAL effector, which binds to DNA. There is a certain part of the protein called the DNA binding domain, which recognizes certain bases in DNA and will only bind to the specific sequence it can identify. This domain is modular, meaning that the binding domain can be engineered to recognize different sequences.
But that actually protein actually is transcription factor consists a DNA binding domain, right? They will bind at different places on the chromosome, and it was found that the DNA binding domain has a modular design built.
Yes.
So like each domain consists about 32 to 34 amino acids, and each domain will recognize or each module, you can see, each module will recognize a particular nucleotide. So then, because it's modular, so you can design a DNA binding domain very quickly to target almost any
Very
kind of
specific.
sequence. Yeah, exactly. So we try to explore that DNA binding domain for various applications, because we can fuse the DNA binding domain with a restriction enzyme, right, like a FOC1 nucleus, and then you can use that one to introduce double-strand break in any position in the chromosome, and then when you introduce the wild type gene, then it can be integrated in the chromosome so that you can correct any point of mutation in the chromosome, or you can actually replace the gene in the chromosome as well.
Entirely.
Yeah, so that's a very powerful tool, and that's clearly is a synthetic biology tool because it's a modular kind of design, right? Because the DNA binding domain, but also the whole artificial nucleus is modular because you can actually have the DNA binding domain, and also you have the restriction enzyme.
So this TAL effector is also attached to a restriction enzyme that can cut through double-stranded DNA. Dr. Zhao's lab uses the TAL effector protein to introduce cuts to DNA at very specific spots where point mutations have taken place. The cut allows access to the mutation so that it can be corrected, completing the goal of gene therapy. This gene therapy approach is a good example of synthetic biology because it uses a gene switch to tightly control when the therapy is delivered, and because the protein itself is modular, or engineerable.